Hello everyone, and welcome to the Medical ITS channel. Today, we are diving into a super important process your body performs every day, hemostasis. So, what exactly is hemostasis? Simply put, it's how your body stops bleeding after an injury. Let's imagine you get a small cut. What happens inside? The body's response unfolds in three main steps. First, the injured blood vessel quickly narrows down in what's called transient vasoconstriction. This helps reduce the initial blood loss. Second, a group of tiny cells called platelets rush to the scene and form a temporary plug. This is known as primary hemostasis. Third, this temporary plug is reinforced with a protein mesh called fibrin, creating a strong, stable clot. This final step is called secondary hemostasis. In this video, we're going to focus on those crucial first two steps, the initial vasoconstriction and the formation of the platelet plug. So, let's start with transient vasoconstriction. The moment a blood vessel is injured, its smooth inner lining, the endothelium, is broken. This exposes the underlying collagen to the blood. Almost instantly, the vessel squeezes itself shut. This happens for two reasons. 1. Nerve reflexes triggered by the pain and injury, and 2. The release of a powerful molecule called endothelin from the damaged endothelial cells. This quick constriction is a lifesaver. It slows down bleeding and, most importantly, buys precious time for the next responders, the platelets, to arrive and get to work. This brings us to our second step, forming the platelet plug. This is how the body creates a quick patch to stop the bleeding. But before we get into the process, let's meet the three main characters in this story: the platelets, a protein called von Willebrand factor, and the subendothelial collagen that got exposed. First up, the star of the show, the platelet. Platelets are tiny, disc-shaped cell fragments floating in our blood. They don't have a nucleus, and they live for only about 8 to 10 days. Their main job: to be the first responders in an emergency. When a blood vessel is damaged, they are the first on the scene, sticking to the injury to form that initial plug. But that's not all they do. They also help amplify the entire clotting process and even play a role in inflammation and tissue repair. A healthy adult has between 150,000 to 450,000 platelets in every microliter of blood. Too few is a condition called thrombocytopenia, and too many is thrombocytosis. Next, let's talk about our second key player, von Willebrand factor, or VWF for short. Think of VWF as the superglue of primary hemostasis. It's mostly made and stored inside the endothelial cells lining our blood vessels, in little packages called Weibel-Pollard bodies. VWF is released in two ways. There's a constant, low-level release that keeps normal amounts of VWF in our blood. Then there's a stimulated release, which happens during an injury, under stress, or even with certain drugs. So what does this VWF superglue do? First, it helps platelets stick to the injury site by acting as a bridge between the platelets and the exposed collagen. Second, it acts as a bodyguard for another important clotting protein, factor VIII, protecting it from being broken down too quickly. It forms these ultra-large, extra-sticky chains that are perfect for catching platelets and starting the plug. Just remember, those endothelial cells are the main source of the VWF circulating in your blood. Alright, now that we know the key players, let's walk through the 4 steps of primary hemostasis. These are platelet adhesion, platelet activation and secretion, platelet aggregation, and finally, interaction with the wider coagulation cascade. Let's begin with step 1, platelet adhesion. This is all about getting the platelets to stick to the site of injury. When the subendothelial collagen is exposed, platelets grab onto it using special receptors on their surface. Think of them as tiny hands. The first set of hands, GPIA, anchors the platelet in place. The second, GPIB, tethers the platelet from the flowing blood with the help of von Willebrand factor. And the third, GPVI, is the main receptor that gets the platelet really excited and activated once it binds to collagen. Now, what happens if these receptors don't work correctly? There's a rare condition called Bernard-Solier syndrome where the GPIB receptor is defective. Without it, platelets can't tether properly, leading to bleeding problems. We can test for this with a lab test using a substance called ristocetin. In a healthy person, ristocetin makes platelets clump together. In someone with von Willebrand disease, they don't clump, but adding normal plasma fixes the problem. In Bernard-Solier syndrome, however, they don't clump, and adding plasma doesn't help because the platelet's own receptor is broken. Once a platelet is stuck, it's time for step 2, Platelet activation and secretion. A stuck platelet is an activated platelet. It changes shape, becoming spiky and more effective at grabbing onto other platelets. It also releases the contents of tiny storage granules. The dense granules release chemicals like ADP, serotonin, and calcium, which call more platelets to the scene. The alpha granules release other important factors that help with clotting and inflammation. This brings us to some important receptors on the platelet surface. The P2Y1 receptor, when activated by ADP, causes the platelet to change shape. The P2Y12 receptor, also activated by ADP, is crucial for sustained clumping. This P2Y12 receptor is actually the target for famous antiplatelet drugs like clopidogrel or Plavix. By blocking this receptor, the drug prevents platelets from aggregating too much, which is useful in preventing heart attacks and strokes. So to quickly recap the receptors, GPIB is for tethering to VWF, GPIA is for anchoring to collagen, and GPVI is for the main activation signal from collagen. After activation, we move to step 3, platelet aggregation. This is where the platelets all stick together to form a plug. The key to this step is another receptor called GPIIB-IIA. When a platelet gets activated by signals like ADP, collagen, or another powerful substance called thrombin, this GPIIB-IIA receptor changes its shape. This change allows it to grab onto a protein called fibrinogen, which acts like a bridge, linking one platelet to another. This is the final common pathway for platelet aggregation. No matter what starts the process, it all comes down to activating GPIIB-IIA. And this receptor is also a major drug target. There's a rare genetic disorder called Glanzmann thrombasthenia where this receptor is missing or defective, leading to severe bleeding. There are also powerful intravenous drugs that block this receptor, used in hospital settings to prevent clots during procedures like heart catheterizations. So, there you have it. From the initial vasoconstriction to the platelets adhering, activating, and aggregating, we've built a primary platelet plug. This plug is the body's rapid response to stop bleeding, and it all happens in just a few minutes. It's a beautifully coordinated dance of cells, proteins, and chemical signals. Thanks for watching our deep dive into primary hemostasis. If you found this video helpful, please give it a thumbs up, and don't forget to subscribe to Medica ITS for more clear and simple medical explanations. See you in the next video.